Hello, my name is Libby Longtain, and I'm an undergraduate environmental science major, currently enrolled in Principles of Ecology here at Northern Michigan University. For my research project this semester, I investigated how agricultural cover affects the relative abundance of coyotes, bobcats, and eastern cottontails in Oklahoma, using data from Snapshot USA. I chose these species an environmental factor to better understand how wildlife species interact and how agricultural areas, if at all, disrupt these interactions and influence population dynamics. Coyotes were of particular interest because they have expanded their populations in response to the decline of wolf populations, a trend that is largely due to urbanization and human development. The fact that coyotes have thrived in human-dominated landscapes is intriguing, as it is uncommon for wildlife to flourish alongside human populations. In contrast, bobcats have smaller population sizes and home ranges when living near humans, although they tend to prefer agricultural areas over urban environments. Similarly, eastern cottontails also prefer agricultural spaces, particularly during the growing season, where they can use crop cover to protect themselves from predators. These three species interact frequently, particularly coyotes and bobcats, which compete for eastern cottontails as prey. The goal of my research was to explore how agricultural cover influences these species' relative abundances and their interactions. To achieve this, I used a subset of camera trap data from Snapshot USA in my study area of Oklahoma. I calculated the relative abundance of coyotes, bobcats, and eastern cottontails in areas with above average and below average agricultural cover. Specifically, I measured the number of species detections per 100 trap nights at my Snapshot USA site and performed a t-test to determine if the relative abundance of these species varied between the two types of sites. My research revealed that the relative abundance of eastern cottontails and bobcats did not differ between sites with high and low agricultural cover, while coyote relative abundance did show significant variation. These findings did not support my hypothesis that agricultural cover would decrease the relative abundance of bobcats and increase the relative abundance of coyotes and eastern cottontails. Several factors may explain why agricultural cover did not influence bobcats or eastern cottontail populations. For bobcats, habitat fragmentation or a lack of adequate space could be a limiting factor, while eastern cottontails might not be able to locate food, particularly in the winter when there are fewer crops. In contrast, the increase in coyote populations may be due to the relative absence of wolves in agricultural landscapes, providing coyotes with more space and fewer competitors or threats from larger predators. Agricultural cover can also provide protection from coyote hunters, which would help boost their populations. Further research should investigate the specific mechanisms behind the positive relationship between agricultural cover and coyote abundance. For example, studying the availability of food sources, the competitive dynamics with other predators, and the role of space for dispersal could provide further insights into why agriculture benefits coyotes. Additionally, this research could inform wildlife management practices, particularly whether large coyote populations in agricultural areas negatively affect other species, and whether coyote populations need to be managed to protect biodiversity. Finally, I would like to extend my sincere gratitude to the Snapshot USA collaborative team for granting me access to their invaluable dataset, and to my TA, Ren Rooney, for their guidance throughout the course of my project. Thank you for your time, and I wish you a wonderful day.